Thank you, everybody, for being here. So, deciphering climate-mediated adaptation in European sheep. Climate change has been a hot topic for some time now. Here is a rough estimate of the interest of the general public on climate change, which I modeled using Google Trends data for the query effects of climate change. Well, surely there has been a growing interest, although with up and downs. And if you were wondering the reasons for those ups and downs, I did too, and I think I identified one possible contributor for the latest increase in climate change awareness. So such growing interest comes from a good reason, which is that change will be relevant for all of us. It seems the overall direction of these changes points to a more extreme environments, so hotter climate overall, and either wetter or drier climates. So in the meantime, human population is growing rapidly. A consequence is the increased global demand for livestock production, which is expected to double by 2050. Problem is, on the one hand, we have an increasing request for animal products. On the other hand, we have a changing climate, which has the likely effect of reducing livestock production through competition for natural resources, quantity and quality of feeds, livestock diseases, heat stress, and biodiversity loss. So the challenge is indeed to try and maintain a balance between productivity, household food security, and environmental preservation. From the genomics point of view, what we can do is to identify genomic variants, which can provide advantages in stressful climatic conditions. Luckily, we do not need to reinvent the wheel here, as we can source from the selected variants which brought a livestock species as sheep to successfully thrive in all continents under the most diverse climates. So that's exactly what we explored with my colleagues at Università Cattolica del Sacro Cuore in Italy. And importantly, this project is part of IMAGE, which is an H2020 project coordinated by Michel Tissier-Bouchard, which included many countries, mostly from Europe. And the project itself covers several objectives, all related with the use of genomic information in livestock species. But our group at Università Cattolica del Sacro Cuore focused, as I said, on identifying genomic variants involved with adaptation to climate. So we did use a landscape genomics approach to perform this task. Now, a fundamental mantra in, let's say, science is that your analysis would be as good as the quality of your sampling. So in the specific case of association analysis as landscape genomics, you need to sample both genotypes and environment. So we were particularly careful in balancing the sheep sampling in order to intercept as many climatic clusters as possible. So what we did was to divide the sampling area, which is represented here, using 50 times 50 kilometers grid. And we aggregated the values of 20 environmental variables via cluster analysis. So by doing so, we identified 8 main clusters and several subdivisions of them. So we then guided the animal sampling in such a way to evenly sample all the different clusters. And here is Oliver, which contributed in this bit of the project. Along with the 19 bioclimatic variables plus elevation, we also included the temperature humidity index, or THI, which is a combined index that can help in describing heat-derived stress. So here you can see that we can divide THI in category risks. So heavily stressed sheep here and relaxed sheep here in green. So clearly our analysis, for our analysis, we selected this bit of the graph. So those THI values ranging from green to red, so the most extreme. Now, I just want to point out, if you look at the map, you might appreciate that despite the overall north-south gradients, which was expected, of course, several areas contain more than one category risk. Here you see a green spot. And this is Elia, which computed the THI index for us. So we had the genotypes and we had the climatic variables. We then use LFMM to perform the association analysis between them. So LFMM estimates the influence of population structure, including a specific number of random factors, or latent factors, as they call them. This is quite convenient, whereas without the software, the user needs to provide the random variables to correct for population structure. Only thing is that you still need to suggest how many Ks LFMM should consider. Ks would be the number of latent factors. So to do that, we ran an SNMF analysis and solved for the lowest scoring cross entropy values. And we ended up choosing K19, 23, and 28 for our LFMM analysis. So we performed the analysis and we actually tested each of the 19 bioclimatic variables plus altitudes plus THI for those 3 values of K, so many analyses. However, as we just started to explore the results, today I will just show you what we got at K23 for the THI variable. And this is the result in Manhattan plot. So as you see, we got some p-values popping up from the background. And specifically, we identified 7 genomic regions harboring 12 genes, which resulted significantly associated with THI. So in this first region here, well, the content, the genes within this region were already described to be associated with fat versus thin tail differentiation in sheep. And this is particularly interesting because the fat tail is actually considered an adaptive response to a harsh environment. It is a valuable reserve for sheep during periods of food shortage. Whereas this region here includes a gene, which is GSK3A, which has been associated with response to heat. And here, this region, which by the way was the one with the highest significance, which showed the highest significance, this region has been associated with the regulation of food intake, energy expenditure, and body weight in response to metabolic stresses, which are among the consequences of living in extreme climates. So as per the remaining genes, we are still working on understanding their role. So to move forward, here we attempted something else, something different to extract meaningful information related to climate adaptation. That is, we sought for selective sweeps. Now, the issue is that with LFMM, whatever we fished out as significant, well, that meant that it was significantly associated with a specific variable. So to the best of my knowledge, this is something you cannot do explicitly when you perform selection signature analysis. Hence, we conceived a little framework to maximize the selection signals putatively related with environmental adaptation, while reducing the signals involved with species differentiation. What we did is exemplified, or surely oversimplified in this slide. So the idea is that for 2 related breeds which developed in the same environment, so here and here, the selection analysis will mostly pick up those signals differentiating the breeds, but shouldn't as easily identify environment-driven selection, as the 2 breeds likely underwent parallel selection for that specific selective pressure. So If we identify the signals due to the species differentiation within a same environmental cluster, we could then sort of subtract them from the selection sweep we identify on the whole pool of individuals and environments. So to identify selection sweeps, we used HAPFLK, which is very nice as it corrects for allele frequencies for population structure, sort of discriminating selection from drift. And to try maximize the environment-driven signals, it was very important that we selected those individuals experiencing the most extreme climatic conditions. As an example, here, to test for adaptation to temperature, we selected those individuals living in the hottest here and the coldest environment. So it does not surprise that we selected a lot of breeds from North Africa and Scandinavia, of course. Then we used the same approach to test dry versus humid environments. And also we did the same for the THI index. So, and here are the HAPFLK results for the hot versus cold contrasts. In tones of gray are the selection results obtained for the full set of individuals, hence experiencing better both extreme cold and extreme hot climates. In blue are the top HAPFLK signals obtained using only the individuals living in extreme cold climates, so blue, and in red, the top signals from those living in extreme hot environments. So as you can see, most of the peaks are somehow shared among all the individuals, irrespectively of the climate they live in. However, we got some peaks which show no red-blue signals, and then our suspect is that they might be due to climate adaptation. And we did the same for humidity, and we did the same for THI, and you see that in both cases, we got candidate regions. So then we wrote down which genes were harbored in those genomic regions of selection, and we identified quite a bit of genes using this approach. And we are now in the process of doing some literature mining on them. Interestingly, but I would say not at all surprisingly, several genes were common among the THI, hot, cold, and humidity tests. They are strongly correlated with each other. We're starting with the three genes in common among the three tests. We found that these two regions, which have been previously associated with heat tolerance and this very general definition of climatic stress, so straight on point. And just to move forward a bit, if we look at the genes in common between the hot, cold, and a humidity test, so here, we find that all these well-known genes, which seems to play a crucial role in various adaptation to climate, really. These results are in fact quite comforting, as they tell us that our rationale to intercept selection due to adaptation to climate was not that bad, albeit simple maybe. So just to wrap up, we identified several genes, many already known for their involvement with climate adaptation. And that's good, as our approach using both alpha-MEM and HAPFLK seems to work fine. But of course, there's much more we need to do, and namely, we still need to explore the LFMM results obtained for each environmental variable. And that's going to be long and tedious, obviously, but still, it's going to be really interesting to see what comes up. And also, we do need to explore the putative adaptive significance of all those genes which we identified but have not been previously described. So, and that's going to be a challenge. Maybe for the most interesting one, we will try and attempt some lab tests, and see if we can replicate their function. And so I would say, yeah, this is basically where we are at the moment. And what is left to do is just to acknowledge all the people that is working on this project. So let me thank you, all the colleagues which are closely involved with the research, which are the people at the Aimon Emerson lab at Università Cattolica del Sacro Cuore. So, of course, Paolo Aimon, Marsan Licia-Colli, Lisa Sonenzi, and the colleagues from APFL and PTP, Stefan, Elia, Olivier, which helped a lot, did a lot of analysis with us, and of course, Alessandra Stella and Barbara Lazzari and Marcello Del Corvo. And well, here are all the partners of the image projects, which are way too many to name them individually. So, well, and of course, all of you, I need to thank you, all of you, for your attention, and thank you, and I'm happy to answer questions.